dear students, I welcome you in my channel Eduscope. Today I am going to start the new chapter Biotechnology. This chapter Biotechnology is so vast, so I divided it into different videos. I am going to start the introduction part of this biotechnology. First of all, biotechnology means what? It is a branch of biology which deals with the technique of using living organisms or their products like enzyme from organisms to produce products and processes useful to human is called biotechnology. So it is a branch of biology where the living organisms, most of the cases microorganisms, either whole body or their product are used for human being. That is biotechnology. Simply we can tell that the curd which is produced from the milk, it is the product of biotechnology. It has been used for a long time in our society. The term biotechnology was coined by Carl Erecke in the year 1917. So simply we can tell it, the biotechnology means the use of microorganisms or their products and which are very much helpful for all human beings. Biotechnology is a very vast subject. It includes microbiology, biochemistry and genetic engineering. It has been divided into two groups. One is traditional biotechnology and another is modern biotechnology. Traditional biotechnology means what? What we use in our home. Very easily we can get the product like curd or wine. But modern biotechnology includes the development of test tube baby, DNA vaccine and synthesis of recombinant DNA. So these are the example of modern biotechnology. There is an organization, European Federation of Biotechnology, EFB, which controls all research work what is being done in different parts of the world. Now I am coming to the principle of biotechnology. Actually, it has two core techniques. Number one, genetic engineering, and number two, maintenance of the contamination-free condition, that is sterile condition. Actually, genetic engineering means what? We can change the nature of the gene and that should be incorporated into the host cell which gives us our desired product. That is genetic engineering. Actually, we can engineer or we can change the structure of the gene which is called recombinant DNA, and this has to be introduced into the host cell to get our desired product. Father of genetic engineering is Paul Berg, and in the year 1980 he was awarded Nobel Prize. This Paul Berg introduced SV40 gene into a bacterium with the help of lambda virus. Second principle is that maintenance of the contamination-free condition. That means what recombinant DNA is produced by the process of genetic engineering, that should be allowed in such a condition where this host having this recombinant DNA can grow very fast and gives our desired result. Now question is, why this genetic engineering or recombinant DNA technology is very important? We know that all living organisms reproduce. First, very simple type of reproduction is asexual. In asexual reproduction, what the organisms are produced, that are called clones, because they are all similar, their variation does not occur. After that, in sexual reproduction, variation occurs by crossing over. Now, next process comes hybridization. In this process, the cross takes place in between two breeds. In between two breeds with the good qualities, when they are crossed, what offsprings are produced? In this offspring, some desired character is also there, and along with undesired characters is also there. Undesired and desired, both types of characteristics are present in the offspring during this process of hybridization. Next comes genetic engineering. During genetic engineering, we can change the structure of the gene by recombinant DNA technology. This DNA structure is changed and that are incorporated into the body of host with the help of some vector. Vector means which carry this gene into the host, and this host produces some product, and these products are useful for human being. So this is the most modern technique, the genetic engineering. According to the requirement of the scientist, this change gene with the help of vector transported to the host, and this host, when these are cultured in they are a good medium, they are producing the products, and these products are useful for human being. For this process, biotechnology, we have to make the recombinant DNA. That means the changed DNA. This change has been done according to the will of the scientist, according to the requirement of the scientist. Now, here some key tools are required. The key tools are required for this are recombinant DNA technology. What are they? First of all, source, from where we have to collect the desired gene. From where we have to collect the desired gene, that is the source. Next, this gene should be cut with the help of an enzyme that is called restriction enzyme, which is also called molecular scissor. It cuts like a scissor. Next, polymerase enzyme. This polymerase enzyme is very much helpful for the process of polymerization. Next is ligase enzyme. What portion is cut? That should be attached with the vector's DNA. That is why this ligase enzyme is called molecular glue. Next comes vector. Vector are nothing but a carrier of this desired gene into the host body. And at last, the host, the desired gene, will produce the products for human being. So for this recombinant DNA technology, these are the main steps. These are the main steps for recombinant DNA technology. We have to learn it every points. We have to learn every points of it. There are 10 points, and we'll learn step by step every one in detail in my next video. First of all, the selection of the desired characters. Yes, the scientist has to find out which one is the desired one, on which character they have to work post. That is the number one, selection of the desired characters. Next is that characters containing DNA should be isolated. That character containing DNA should be isolated. So isolation of DNA that having desired characters. This is the point number 2. After getting this DNA which are having that desired character, we have to fragment it. Fragmentation of DNA by restriction endonuclease. You know, this restriction endonuclease acts as molecular scissor and cuts the DNA particle into small unit or small fragments. Next comes isolation of the desired DNA fragments by gel electrophoresis. Very important process, that these smaller fragments should be isolated. The bigger fragments and the smaller fragments should be differentiated by this process of gel electrophoresis. It's a very unique process and very important process for this recombinant DNA technology. Next comes what small portion is getting by this gel electrophoresis, that should be amplified, means making many copies of it, means many copies to be formed by the process of PCR. PCR means polymerase chain reaction. Polymerase chain reaction. It is also very important steps for this recombinant DNA technology, because this process amplify the DNA fragments. Ligation of DNA fragments into the vector. Yes, I told you, when the DNA of the vector and the desired DNA are fused together by the process of ligation by the enzyme ligase that forms recombinant DNA. And this recombinant DNA to be inserted into the host cell. So this point tells insertion of recombinant DNA into the host. Next comes culture the host cells in a suitable medium. Yes, what host we obtain, that should be grown in a suitable medium on a large scale. On a large scale, we have to culture them in a suitable medium. That is point number 8. After point number 8, point number 9 tells the extraction of the desired gene product. What gene has been incorporated in the host cell, they will produce some products, and these products should be extracted. That is point number 1, extraction of the desired gene product. Last process is downstream process. In this process, what products we have been extracted, and these are to be purified and are to be made ready for marketing. That is called downstream process. In my next video, I'll discuss every point of this in detail. So if you want to see the upcoming video, you please subscribe my channel Eduscope. Thank you very much for being with us for a long time.